Hello students, welcome to EPG Pathshala. I am Dr. Sanjukta Subuddhi, Senior Fellow working in the Department of Environment and Industrial Biotechnology, the Energy and Resources Institute, New Delhi. Today we will learn about the module Microbial Biohydrogen Production under the paper Environmental Biotechnology. The objectives of this module is the importance of biological hydrogen production process. We will discuss what are the significances of different processes of hydrogen through biological route. In addition to that, we will also learn the life pathways followed by the microbes through biological route to produce hydrogen, and difference of these pathways, the biocatalysts involved in catalyzing the hydrogen production through these processes. We will also discuss significance and limitations of these biological processes, including future prospects of this process. Before learning in detail about the microbial biohydrogen production processes, I would give a brief introduction about the significance of hydrogen. Hydrogen has been seen as one of the most promising energy carrier because of the fact that it has a very high energy content, which is around 2.75 times higher than the energy content of a hydrocarbon fuel. Though it has a very high energy content, it cannot be used as directly as an energy source. It is an energy carrier and needs to be converted to a form of energy such as electricity, which can be generated by passing that hydrogen through the fuel cell. Hence, this has got very good significance to use as an automotive fuel. And other significance aspects of this hydrogen is this is CO2 neutral. When it burns, it doesn't release any greenhouse gases. Hence, this is a very clean form of energy and carbon neutral and thus environmental friendly. Globally, there is a high demand for hydrogen. Presently, mostly the industries, refineries and fertilizer industries, they use hydrogen in a large scale for production of fertilizer, the ammonia, and processing the crude oil. Hydrogen demand is being seen to escalate by around 3.5% through 2008 to around 290 billion cubic meters. This is the schematic diagram of a fuel cell based vehicle, where we can see here hydrogen can be converted to the form of energy, that is electricity, by passing through the fuel cell, and that can serve as the source of energy for driving this vehicle. Hence, hydrogen has got significance to use in the automobile industries as a source of energy. As we have learned in our previous lecture, gaseous biofuel, hydrogen is one of the significant energy carrier, and presently mostly hydrogen is being used for the production of ammonia by the fertilizer industries and by the refineries for processing the crude oil. So current demand of hydrogen is being met by producing hydrogen through physicochemical processes. The significance of these processes is that the cost of this hydrogen production which is produced from these processes. However, the constraint of these processes is these processes rely on the use of finite fossil fuel sources. Hydrogen through these processes is mainly generated by partial oxidation of heavier hydrocarbons and coal, and also by the steam reforming of the lighter hydrocarbons, and other process is the thermal cracking of natural gas. Major amount of hydrogen demand is being met by production of hydrogen through these three processes. The limitation of these processes is that these are very highly energy intensive, And apart from that, production of hydrogen through these processes emit large amount of greenhouse gases. So the concern over here is the climate change. And apart from that, the requirement of input energy. Out of 100% of required hydrogen, almost more than 95% of hydrogen is being produced through these processes. Only very few amount, that is 4 to 5% of hydrogen, is produced by electrolysis of water. So the global attention is towards for production of hydrogen from renewable sources or from the alternative sources which are not associated with the emission of greenhouse gases. So in this context, if we see here, the biological processes for hydrogen production have lot of significances compared to these thermochemical or physiochemical processes. Biologically, hydrogen can be produced mainly by four processes, as we have learned in our previous lecture. One is the dark fermentation process, one is the biophotolysis, the other is the photofermentation process, and the number fourth is the integration of two processes, that is photofermentation process with the dark fermentation process. In terms of significance of these biological processes is that these processes do not require high input energy because they can be operated in mild condition and ambient pressure, and on the top of that, carbon dioxide or any greenhouse gases is not released from these processes. Hence, these processes are considered as environmental friendly. So we will learn in detail about different biological processes for hydrogen production and the pathways associated with production of hydrogen and the type of microbes they have got potential for production of hydrogen. Biophotolysis is one of the process. As the name indicates over here, photolysis of water occurs by the microbes through the biological process. Few of the algal and the cyanobacterial strains like spirulina, chlorella and chlamydomonas, they have got this potential to produce hydrogen by photolysis of water, hence the name is biophotolysis. As we can see from this slide, these microalgal strains, they use water as the source of energy and convert that to hydrogen and oxygen, and this process occurs in the absence of oxygen because these enzymes are highly sensitive to oxygen, and hydrogen production processes are normally occurred in the anaerobic condition. The enzyme that serves as a biocatalyst for production of hydrogen here is the hydrogenase. The significance of this process is the use of water as the source of energy and production of hydrogen, and this process does not lead to the production of any other greenhouse gases. The limitations of this process is the production of oxygen. As I have mentioned here, the enzyme which catalyzes the production of hydrogen here is highly sensitive to oxygen. So the oxygen which is released from this process tends to inhibit the catalytic activity of this enzyme. So the major challenge of this process to how to scavenge the oxygen which is released from this process. Though there are many significances, but the challenge is the simultaneous release of oxygen and the requirement of light as the source of energy for this reaction, catalytic reaction. Hydrogen production pathways differ in different microbes. In terms of biophotolysis process, these processes are again two different types. One is direct photolysis and one is indirect photolysis. The green algal strains, mainly the Chlamydomonas reinhardti, chlorella, they encompass the pathway for direct biophotolysis process, where the hydrogenase enzyme in presence of light energy carries out this catalytic process of water to produce hydrogen and oxygen. And the light energy wavelength requirement for this process is lower than the 680 nanometer, which is mainly absorbed by the photosystem II. While in terms of indirect biophotolysis, in case of indirect biophotolysis process, which is mainly carried out by the cyanobacteria such as Anabaena variabilis, the hydrogen production occurs from water through two different steps here. In the first step, in presence of light energy, water first converted to the glucose and oxygen, and in subsequent step, carbohydrate in presence of water and light energy converted to hydrogen and CO2. The significance of this process is the production of oxygen occur in a separate compartment and hydrogen in a separate compartment. So the oxygen which is released doesn't tend to inhibit the activity of hydrogenase simultaneously, but the constraint is how to manage effectively production of hydrogen by controlling two steps in a timely manner. The significance of these biophotolysis processes is the requirement of water, and that's why these processes are very clean. However, to make these processes commercially viable, many challenges need to be addressed. Similarly, when we move to the dark fermentation process, as the name indicates, dark means this process doesn't require light as a source of energy, and hydrogen can be produced in the absence of light through the fermentation process from different organic carbon sources. Few of the microbes like Clostridium, Enterobacter, Citrobacter, Bacillus species, these microbes, they have got the potential to produce hydrogen by fermentation of the organic sources, carbon sources, in the absence of oxygen. As we have learned earlier also, biohydrogen production process is a strictly anaerobic process, and these processes occur in the absence of oxygen. As we see over here, different microbes like Clostridium and Enterobacter, they produce hydrogen by following different pathways. And the hydrogen yield efficiency is maximum of 4 mole of hydrogen per mole of glucose for the Clostridium species, that is again related to the pathway associated with hydrogen production by these microbes. We will learn in detail the pathways and the metabolites those are produced during this process. However, when we compare to the hydrogen yield efficiency of Enterobacter microbe, the pathway that is followed by these microbes, the efficiency of hydrogen is less, is around 2 mole of hydrogen per mole of glucose, and that varies when the starter material is a C5 sugar. The significance of this process is the hydrogen production rate is very high compared to the other biological processes. However, the limitations are there, like only hydrogen is not produced from this process. This process is accompanied with the production of some of the gaseous as well as the liquid metabolites. Hence, the overall hydrogen yield efficiency from the substrate or the feedstock is less. And another advantage of this process is that the waste organic sources can serve as a good source of energy for production of hydrogen by these microbes through this process. As we have learned that the dark fermentation process is being carried out mainly two different type of microbes, that is the Clostridium and the Enterobacter. The type of pathways also varies in these groups. As we see here, the typical pathway that is being followed by the strains of Clostridium for production of hydrogen from glucose, the maximum hydrogen yield efficiency through this process is around 4 moles of hydrogen per mole of glucose. The soluble metabolites which are produced during hydrogen production through this dark fermentation process in this pathway are mainly can be of the short chain organic acids like acetic acid, butyric acid, and the simple solvents like the ethanol, butanol, and acetone. The yield efficiency of this process, hydrogen yield efficiency, varies with respect to the type of metabolites generated. It was observed that maximum yield efficiency, that is a 4 mole of hydrogen, can be generated by these microbes through this process when only acetic acid will be the metabolites of this process. The yield efficiency goes down when the production of other metabolites also occur through this process. The hydrogenase enzyme, which serves as the main biocatalyst for hydrogen production in this process, are mainly the iron hydrogenase enzymes. These microbes are very strict anaerobes. This process occurs in anaerobic condition, like we learned previously for this process. Coming to the mixed acid fermentation pathway, which is mainly carried out by the group of enteric bacteria like Enterobacter. Here, production of acetic acid, also butyric acid, is being observed, and some of the strains, they follow the pathway to produce hydrogen that generate some of the solvents also as one of the liquid metabolite like ethanol. The difference between these two type of microbes is that these microbes are the facultative microbes. They can survive in the presence of oxygen. However, hydrogen production by these microbes occur through anaerobic process only. The type of hydrogenase enzyme that is being involved for catalysis of this process in these microbes is mainly nickel iron hydrogenase. However, in terms of the pathway which we discussed for the Clostridium, the hydrogenase enzymes are mainly iron-iron hydrogenase enzyme. The catalytic potential of iron-iron hydrogenase enzymes are many-fold higher than the catalytic activity of nickel-iron hydrogenase enzymes. Compared to the yield efficiency of this enteric bacteria, maximum of 2 mole of hydrogen can be obtained theoretically from 1 mole of glucose, which is less than the hydrogen yield efficiency that is being observed from the Clostridium groups of bacteria, which encompass iron-iron hydrogenase as the biocatalyst. Coming to the photofermentation process, as the name indicates here, this process requires light as the source of energy, and through the fermentation process, in presence of light, hydrogen can be produced. But the type of microbes having the potential for production of hydrogen from this process is different compared to the other two processes which we have learned earlier, that is the biophotolysis and the dark fermentation process. Only few of the bacteria such as Rhodobacter spheroids and the Rhodosporomonas belonging to the group of purple non-sulfur bacteria, also known as the photosynthetic heterotrophic bacteria, they follow a different pathway that utilizes short-chain organic acids and some of the carbohydrates. That again varies the type of pathway involved by these microbes and produce hydrogen in presence of light, and they use light as a source of energy. Again, this process also occurs in anaerobic condition in the absence of oxygen. As we are seeing that different pathways are involved for the production of hydrogen by these microbes, these processes require the use of different enzymes or the biocatalysts. The enzyme involved over here is the nitrogenase enzyme. Unlike the enzymes involved for the hydrogen production in other processes are the hydrogenase enzyme. Since the nitrogenase enzyme is involved here for production of hydrogen, this process occurs in the absence of nitrogen under the nitrogen stress condition. So, in presence of light, in the absence of nitrogen, this process occurs in the presence in anaerobic condition where argon gas serves as the gaseous atmosphere in the headspace of the reactor, and the hydrogen is produced from the organic acids such as the acetic acid. The significance of this process is the hydrogen yield efficiency is very high for this process as compared to the dark fermentation process, and it requires light as a source of energy. But the limitations of this process is that the rate of hydrogen production is very low. Hence, there are many challenges need to be addressed before moving this process to the scale-up level or subsequently to the commercial level. And another requirement is the requirement of anaerobic photobioreactors. Since light is the source of energy, so it requires the large surface area of the photobioreactor for efficient light energy availability to all the cells in that process and absence of oxygen. These two are becoming the major challenge of this process. But the hydrogen which is generated from this process is very pure, and this biogas is composed of at the most 4 to 5% of CO2 only. Around 95 to 96% of this biogas produced from this process is the hydrogen, and thus this gas which is produced from this process does not require any further purification. It can be directly allowed to pass through the fuel cell for conversion to the electricity. In case of photofermentation process, nitrogenase enzyme serves as a backycatalyst for production of hydrogen. The significance of this process compared to the photosynthetic process is these microbes, they harbor photosystem 1. They do not have photosystem 2, and thus in the absence of this photosystem 2, purple non-sulfur bacteria eliminates the problem of oxygen inhibition in hydrogen generation. So there is no oxygen, as we can see from here, no oxygen is generated through this process. And nitrogenase enzyme here uses short chain organic acids as the substrate or feedstock and converts that to hydrogen and CO2, and CO2 is generated in very small amount, around 4 to 5%. Many of the short chain organic acids can be utilized by this bacteria for hydrogen production, such as acetic acid, lactic acid, malic acid, and only few of the strains of this PNB bacteria, they use the carbohydrates, the simple carbohydrates like glucose directly for production of hydrogen. However, the hydrogen yield efficiency varies with respect to the different substrates which are utilized by this bacteria. For example, when acetic acid is used as a substrate, the hydrogen yield efficiency can occur to the maximum limit of 4 moles. In case of lactic acid, maximum hydrogen yield efficiency is around 6 moles of hydrogen per mole of lactic acid. Moving to the malic acid, the hydrogen yield efficiency is around again 6 moles of hydrogen per mole of malic acid. In case of glucose, yield efficiency can go as high as to the 12 moles of hydrogen per mole of glucose. However, the ease of conversion of lactic acid and malic acid is observed to be more significant for hydrogen production practically by these microbes compared to the other short chain organic acids. The another process is the integration of two processes which we learned before, that is dark fermentation process and photofermentation process. This integration is possible considering the synergistic usage possibility of the spent effluent which is released from the dark fermentation process as feedstock for the photofermentation process. The interesting parts over here is the dark fermentation process releases many of the metabolites, the liquid metabolites as well as the gaseous metabolites, like I have explained before. The gaseous metabolites are CO2 and some amount of methane and H2S, but most of the cases that can be controlled by diverting only for the production of one metabolite, CO2, along with the hydrogen. The liquid metabolites released during this dark fermentation process are mainly the acetic acid, butyric acid, in some cases lactic acid, and some of the solvents like the ethanol, acetone, butanol. These metabolites again vary with respect to the type of pathways followed by those microbes to produce hydrogen. Why the significance I am highlighting here is there. The organic acids which are released as the liquid metabolites during dark fermentation process can very well be served as the feedstock or the substrate or the source of energy for production of further hydrogen by photofermentation process through the use of purple non-sulfur bacteria. As we have learned from our previous slide, the purple non-sulfur bacteria uses the short chain organic acids. Hence, so there is a good possibility for integrating the photofermentation process with the dark fermentation process. By integrating these two processes, the waste generated from the first process can be very well treated for further energy recovery in the second process. The energy could be in the form of hydrogen. So the overall hydrogen production from the starting substrate of the dark fermentation process can be increased. And by integrating these two processes, maximum hydrogen yield efficiency of 12 mole of hydrogen per mole of glucose can be obtained, which is theoretically possible. These are the main advantages of this process. The disadvantage of this process is like the process we have explained before, that is the requirement of photobioreactor in the second process, and the rate of production of hydrogen is low here. So there is a requirement to address these challenges to make this integration process commercially viable. Among different biological processes of hydrogen production which we have learned in these previous slides, the maximum attention is being given to the dark fermentation process. The reason, the rate of hydrogen production from this process is high, and this process can make use of the organic waste from different industries as feedstock for hydrogen production. So that can help in achieving the dual process of waste treatment along with the energy generation in the form of hydrogen. Hence, scientists globally focused more on research explorations for production of hydrogen through dark fermentation process. Leads are already obtained at global scale for production of hydrogen through dark fermentation process in pilot scale from molasses like sugarcane molasses, beet molasses, synthetic carbohydrates, simple sugars. However, presently the concern is for the use of non-feed competitive feedstock because molasses, considering the concern for food security along with the energy security, the global attention diverted towards production of biofuel from next generation feedstocks which do not rely on the food. Hence, for convenience, biofuel production processes are categorized into different generations. One is first generation biofuel production, which rely mostly on the feedstocks that are feed competitive or food competitive. However, considering this food security, the attention diverted towards production of the non-food competitive feedstock. Hence, importance given recently to the second generation biofuel production. Second generation biofuel are those fuel which are produced from the non-feed competitive feedstocks such as lignocellulosic biomass like rice straw, wheat straw, sorghum stover, sugarcane bagasse, sugarcane trash, many of the aquatic plant biomasses. Similarly, the term third generation biofuel is given to the production of oil from the algal based biomass. The fourth generation biofuel are those biofuel which are produced from the genetically modified algal strains. These terminologies are given based on the convenience for understanding different biofuel production processes from different feedstocks. Major focus is being given currently for the use of lignocellulosic biomass for production of biofuel. The reason in India, when we see, we mainly depend on the agriculture as our mainly livelihood. More than 80% of population they depend on the agriculture as their livelihood. So there is a huge amount of agricultural waste is being generated annually, which is estimated to exceed around 220 billion tons. If we can tap energy from this huge source of agricultural lignocellulosic biomass, the energy we can generate, which would be equivalent to around 60 to 80 billion tons of crude oil. As we can see, major amount of this lignocellulosic biomass can be used for tapping our energy in the form of biofuel. How? Because this biomass are mainly composed of three major components. One is cellulose, one is hemicellulose, one is lignin. These three components are present in different proportions which vary in the range of 30 to 50%, 20 to 35%, 10 to 15%. The challenge lies in separation of these components from this lignocellulosic biomass, like separation of cellulose and hemicellulose from lignin. And after separation, cellulose and hemicellulose, which are the complex forms of C6 and C5 sugar, can be converted through treatment procedures to the simple sugars of C5 and C6, which can be fermented easily by the microbes as the source of energy. So the fuel or the biofuel which will be produced from this lignocellulosic biomass, which are also known as the second generation feedstock, is termed as the second generation biofuel. If hydrogen is produced through biological route by using this biomass, this will be termed as the second generation biohydrogen. Seeing the composition of the lignocellulosic biomasses, as this biomass is comprised of a large amount of sugars, the carbohydrates, in the form of cellulose and hemicellulose. So these carbohydrates can be tapped as a source of energy or production of the hydrogen, because presently large amount of these biomasses, mainly the sorghum straw, wheat straw, rice straw and sugarcane bagasse, which are generated after post-harvesting of the crops, are being burned by the farmers. That not only creates a concern for the climate change because of the release of the greenhouse gases, but also raises a concern for the wastage of the large amount of energy. But these sugars need to be converted to the simple sugars or the fermentable sugars prior to conversion into hydrogen by using the microbes. Because the hydrogen producing microbes, they encompass the enzymes which uses the simple sugars, and again not all hydrogenous enzymes can take off these C6 or C5 sugars, though most of the microbes can take off C6 sugars, but use of C5 sugar is a major constraint for many of the microbes, because those microbes, they do not contain C5 utilizing enzymes for conversion into hydrogen. As I have explained before, these lignocellulosic biomass samples need to be converted to the fermentable sugars. Three major components are there, as we can see: lignin, cellulose, hemicellulose. Cellulose is the complex forms of C6 sugars. Hemicellulose is the complex form of the C5 sugars. Around 30 to 35% of cellulose and 30 to 35% of hemicellulose are present in the lignocellulosic biomass samples. So these complex carbohydrates need to be treated for conversion into the simple sugars. So the challenge here is the cost-effective treatment of these complex carbohydrates to the simple sugars, so that eventually these simple sugars can be taken up by the hydrogen producing microbes or production of hydrogen. Though many of the technologies are being developed or established for conversion of these complex carbohydrates to the simple sugars, the challenge now is for the cost of these treatment processes. Again, there are two different treatment processes. One is the acid treatment, another is the alkaline treatment. When we treat any lignocellulosic biomass through the dilute acid-based treatment, that leads to the generation of mostly the C5 rich sugar, that is the hemicellulose. So The challenge here to use the C5 sugars as the source of energy or the feedstock for hydrogen production. Only those microbes which have the potential to utilize C5 sugar, they only can take up these hemicellulose components after conversion to the hydrogen production process. So the challenge is the screening of the microbes or domestication of the microbes which is capable of utilizing the C5 sugar for hydrogen production. And to make the overall process cost effective, now the challenge is to reduce the cost of treatment process, the preliminary treatment process of lignocellulose biomass to the simple sugars. Since the food waste feedstock to use as a substrate for energy production is a major concern these days because that compete for the agriculture, and there is a concern for use of non-food competitive feedstocks, and lignocellulose biomass comes under this non-competitive feedstock. This is also known as the second generation bio feedstock for production of energy. Hence hydrogen which is produced from this lignocellulose biomass is also known as the second generation hydrogen. 2G indicates here second generation hydrogen production. There are many of the leads being obtained on production of hydrogen from this lignocellulose biomass. However, most of these are confined to the laboratory scale and few are at the pre-pilot scale. This process requires intensive explorations for moving this process to commercialization scale, and main constraint is the cost for the treatment of this biomass which needs to be addressed. So students, let us summarize what we have learned in this module, microbial biohydrogen production. In view of the energy security and environmental concern, hydrogen production through biological route offers a promising approach for clean energy generation, as these processes are not associated with generation of greenhouse gases. Hydrogen can be produced biologically from organic sources, water, short chain organic acids through 4 different pathways which we have learned in our previous slides. These pathways, which are encompassed in few of the unique microbes and algae, cyanobacteria and purple non-sulfur bacteria. Considering the concerns of food security along with the energy security, use of non-feed competitive feedstocks, otherwise known as the next generation feedstocks such as lignocellulose biomass, got importance for production of hydrogen in a cleaner form. There are several challenges associated with these hydrogen production processes. Before moving these biological hydrogen production processes to the commercial scale, it is very essential to address the challenges. So in order to make the overall hydrogen production process cost economically feasible, it is very essential to make the process zero waste discharge. That means the discharge which comes out of the process after production of hydrogen can be very well used as a no cost based feedstock for production of the high value commodities, so that the overall hydrogen production process in a refinery mode can be made cost economically feasible. Thank you very much.